Good afternoon, everyone. I would like to welcome you to the third webinar of the Quality Webinar Series organized by EATRIS. My name is Rosanne Vechter and I'm Education Manager at EATRIS. I will be hosting this webinar today. Today, we are very pleased to have two speakers joining this webinar about reproducibility in preclinical and animal experimentation. Before I introduce our speakers, I want to make you aware that this webinar is being recorded, including questions and answers at the end of this session. The recording will be made available on the EATRIS website shortly after this session. We have reserved some time at the end of this session for questions. We invite you to write your questions down in the box in the control panel, as you can see on this slide. First speaker for today is Björn Gerlach. Björn is an immunologist by training with experiences in immune cell signaling and neuroregeneration. In 2015, he was co-founder of the Partnership for Assessment and Accreditation of Scientific Practice, in short PASP, which operates in three different areas. First one is the operational risk management in preclinical research. Second is trainings on data robustness and reproducibility. And third is participation in public-private projects to increase data quality and research outcome. Björn is Project Director and Chief Operating Officer at PASP. Second speaker for today is Dr. Michael Raas. Michael is biologist and research manager. After his PhD and a postdoc position in the field of behavioral physiology at Max Planck, he changed to the Helmholtz Center Munich as lead project manager for INFRAFRONTIER preparatory phase and head of the INFRAFRONTIER project office. INFRAFRONTIER is the European research infrastructure for the development, phenotyping, archiving, and distribution of model mammalian genomes. In 2010, he received a master's degree in Science Communication and Marketing of the Technical University in Berlin. Since 2014, he is head of general management of INFRAFRONTIER, the management and coordination unit of INFRAFRONTIER research infrastructure. One of his responsibilities is to coordinate the implementation of the INFRAFRONTIER quality management framework. I will now hand over to Björn.
Thank you, Rosanne, for this wonderful introduction. I hope everybody can hear me and can see also the slides now. Very excited to present this EQIPD project, which focuses on the reproducibility in preclinical experimentation. But before we go into the project, I want to go one step back, because for decades or maybe even centuries, we had the self-correction in science, which worked sufficiently. We had on the one hand the discovery, and that was confirmed by other groups or was falsified. But in recent years, it seemed to not work properly, this self-correction, or at least was heavily slowed down. And this was mainly shown by two studies from Amgen, by Berkeley and Ellis, and Bayer Healthcare from Prince, who showed that only 80 to 90% of the work could be reproduced. And this was summarized in this Friedman paper, probably all know, who said that 50 to 90% of the research studies could not be confirmed. And they calculated that the costs are about 28 billion in the US alone. And I would like to go into two more detailed examples now. One of them is this compound NXY059, which was used in animal stroke models. And this research group by Malcolm McLeod, they did a meta-analysis about this compound that also went into clinical trials. And they looked for quality measures like randomization and blinding and dissected whether these research studies use these measures or not. And they actually found a very interesting, or made a very interesting observation that when these measures were introduced, the efficacy was in both cases much lower than when they were not used. So really indicating that there is some kind of research bias. And interestingly enough, also all human trials later on, they failed. And it would have been interesting whether the studies in human would have been done on the basis of these observations. Another very, very devastating example comes from ALS research, where also several hundreds of compounds were tested positive in preclinical trials and also went on to clinics. And here you see in green the survival rate that was published for these different compounds tested in animal studies. So you also see a quite positive effect. But when this ALS Therapy Development Institute used these compounds again in studies with good research practice attributes, they were not able to come up with the same results. In fact, of more than these 100 compounds previously reported to slow down ALS progression, none showed any positive effect when retested under proper conditions. So the question here really is something missing? And because many data from these animal studies are not robust or difficult to reproduce. And up to date, there are no comprehensive or generally agreed upon universally followed guidelines and principles to set these criteria for a rigor in study design and conduct and analysis. There's also limited knowledge about which experimental factors influence the reliability and predictability of these animal studies. And for that, the European Quality in Preclinical Data Consortium was funded to investigate these different items and hopefully provide some kind of shortcut by introducing this validated focus on good research practice into the whole research process. EQIPD is funded by the Innovative Medicine Initiative, which is a joint undertaking by the European Union and by the FPR partners who are funding this kind of research and projects. So what is EQIPD exactly? So it's the first IMI consortium that is completely dedicated on improving preclinical research and set a standard for the preclinical data quality. It's a joint undertaking by the big pharma companies, and in this consortium that consists of 29 partners, we have Janssen, Roche, Sanofi, so also many of these big pharma companies are on board here that are well known. There are contract research organizations and small to medium-sized entities, but there's also academia present. So the Charité in Berlin with Uli Diernagel, Malcolm MacLeod, so many of the important universities working on this issue. And fortunately, we have also scientific associations. Heavily involved is ECNP here, but also one of these external collaborators from ALAC is contributing a lot. So it's a very broad consortium with a diverse knowledge and a diverse interest in data quality, which I think is very important. The project is running for a little bit over 2 years now and will be finalized by the end of September next year. So the vision is to have a positive impact on animal research and also help in this line of the 3 R's to reduce the waste of animals in research. That will hopefully then also lead to enhance the pace of knowledge gain, which ultimately will shorten the time needed to make new drug treatments available to patients by having better preclinical data set that hopefully then will make it through the clinics, or at least the fallout rate in the clinics will be less. There are several tasks here within this project. One is to basically first look into the research publications and see which variables can influence the outcome of preclinical research. And this will be done with a meta-analysis by one work package and also by historical data analysis, which I also find very interesting because these big pharma companies come together, pool their data and analyze them again. And that will then help in the future to introduce these quality standards. The work package that I'm mostly involved in is the development and definition of a quality system and several compounds. And then also another work package aims to validate the feasibility of this quality system. And the focus now in my presentation will be on this quality system and the work we have done here so far. So this system contains 3 phases, and what has to be done is that 18 core requirements needs to be implemented within a research unit. And these 18 core requirements were the result of a working group from this big consortium to define what is important, what is the basic set of items that need to be implemented in a research unit. And part of these 18 core requirements has to be implemented into this phase 1, which is very rigid and sets then the basis. And the idea here in phase 1 is to have really the absolute minimum of research, of standards, of requirements for every research unit. So no matter what kind of research is done, these phase 1 core requirements will apply. And just to give an example, that's documentation, which is important. Then this is a bit more flexible phase where additional core requirements can be implemented, but also user-specific requirements play an important role, because nobody then the research unit themselves will know what's important for them. So that's in phase 2, a very flexible mixture of items that should be implemented. But to complete the system, then in phase 3, then all other remaining core requirements would need to be implemented to have this sustained, refined quality system. We have developed 3 elements to build some kind of framework. And this is the toolbox developed by us. And this is an online tool, which I'm going to present. And then there is the planning tool. That's a software that helps implementing the quality system. And then there is the dossier, which is stored locally or in the research facility where user-specific items can be stored, can be organized. And we first will dive into the toolbox now. This is an online tool. It's more or less built like a wiki, and it contains of 4 categories: the objectives, which are more general items. I've presented a screenshot here with part of these objectives. And then if you would scroll down here, then there would be item number 2, the key processes, which focus on the research or on the quality requirements for the experimental procedure. And then there are the support processes, which are items that support the research and the experimental procedures. And finally, section 4 then deals with the continuous improvement, which is some kind of internal assessment procedures that are briefly described here, to have some kind of feedback loop again, so that we have a continuous process and a continuous improvement of this research facility. In principle, these 4 categories consist of 14 to 38 different items. They are all numbered here so that they are easy to find and refer to, but nobody is expected to read through them like that, because that would be not very, that would not lead to the actual benefit. And would be a waste of time. But it's about picking these different items that are needed in the research unit. One of them can be the generation, recording, handling, and archiving of raw data. And I presented this example here to show you what is behind these items in the toolbox. They are always built the same way, that we have background and definitions, that there is guidance, that there is some kind of pointed out risks by please do not forget certain aspects. And then there are also resources. And another very important item, because we are talking about that and I want to present, is randomization. So again, definition, what randomization is about, why it is needed, and then the guidance presents some kind of almost like a checklist to see if you want to do randomization, what you need to consider, what you can consider, like block randomization, in block sizes, stratification of variables, and so on. And then there's also in the resources, there are different tools presented here, so links that provide help to randomize, like the NC3R experimental design assistant, quick calcs, or also sealed envelope, which I have even used for my in vitro research to randomize some items on a 96 well plate, at least the rows, not every single well, but at least the rows, to hopefully reduce some bias. And this list, because it's online and we have access to, we can discuss it, this list will always be expanded, and if there is any input, then this can be included here. So, and this implementation, as I mentioned before, is guided. It's guided by the planning tool, and that's there we have developed a prototype which is Excel based. And when I saw it the first time, it was developed by some programmer in the consortium from AppV. I didn't really recognize Excel because it starts with these windows that guide you through the first steps of this first phase. And this first step asks whether all tools are ready to use and and are set up. And then there are four more steps, and these are just several questions then to help set this first framework for this quality system. So who will oversee the EQIPQS? Why is the EQIPQS being set up? And then what, where, and when? So what are the timelines? Who should be involved? When does somebody need to be informed? And then step 5, what I've mentioned earlier, are your data traceable? And this apparently is a very big challenge for many research institutions. And that's why this also comes so early in this setting up process, that we provide guidance on how to make the data traceable and findable. So the item generation, handling, and changes of raw data. So these are then the links that directly connect this local Excel tool with the online toolbox. And another link here is this data security item, traceability of data and parsing having impact on data. And then finally, here is a documentation plan. And that's a little bit different because clicking on that item in the toolbox, in this Excel tool, would then open a Word document that is downloaded with the dossier, so that this Word document can be set up and described directly on the research institution that's going to set up this quality system. So we try to build it very easy, accessible, and that there is only very little effort needed to reach maximum gain. 2 items I just want to briefly mention is, which are also important, is the animal research and confirmatory mode research. So confirmatory mode research versus exploratory research is research where additional standards apply. And once these steps are finalized, then we come to an Excel list, and that's Excel how I always knew it, and presenting here the action plan. And this action plan consists of these 6 core requirements that are already set up in this first phase. and then also lists the remaining 12 core requirements. Additionally, it provides also again here the links to the toolbox and also to the documents. There is the possibility to provide some comments here when these items are implemented, and it should also be documented at this one place when it was implemented, so that there is at least some kind of overview what has been done already. And another button, import needs, that there comes this flexibility in, because these 18 core requirements are just the basis, but for several research units, additional standards might apply. And then these additional standards can be imported into this action plan via the import needs button. So the last thing I was previously mentioning is this dossier that is locally saved on your computer. And here we provided this folder structure, which is numbered the same way like the toolbox, so that items can be easily found. It's also, again, these 4 different categories with the objectives, the key processes, and then here the different items can be saved and are easily accessible for any researcher within the research unit. So, and this was already the last item I wanted to show you. So this EQIPD quality system, I've shown you, it consists of these 3 components: the toolbox, the planning tool, and the dossier. And it is currently beta tested with different consortium partners. And the main goal always was to have a flexible, fit for purpose, lean and user-friendly system. And I can only invite everybody to also to try it, to use it and see whether we matched this goal, or then also make suggestions on how improving it. And with that, I want to thank everybody in the consortium, the different work packages, and especially Thomas Steckler and Malcolm McLeod, who are heading this project, Glenn Beckley, one of the advisors, and Shai Silverberg, who support that project. And I hope that this will be indeed a very helpful suggestion for improving research. So with that, I'm going to hand over.
Okay, thanks a lot. So within the next quarter of an hour or so, I will introduce you to InfoFrontier and the role of this research infrastructure in improving the reproducibility in preclinical biomedical research involving animals. And in this presentation, I will pick up several issues that have been mentioned in this excellent presentation of Björn that we just heard from the perspective of InfoFrontier and from the research infrastructures. Yeah, with this introductory slide, I think this is the basic concept of the entire webinar series, that we want to cross this valley of death and really contribute to a better translation of basic biomedical research into the clinical setting and into the clinical trials. And if we talk about animal research, there are several different reasons why the animal experiments do not translate. One of them could be shortcomings in the process. So on the clinical side or on the clinical part of this valley of death, but this is not the topic of this presentation or this webinar today. So if you go to the side of animal experimentation, of course it could be, or there's cases of inadequate animal data and also over optimistic conclusions that are drawn from animal studies. As in other areas, we have the problem of non-reporting of results or of negative results, so the publication bias, and the general question that results from animal experiments may simply be not transferable to humans. If you look a little bit more into this this area of let's say the area that has also been touched or that the EQIPD project wants to improve. So there's many items, and it's surprising to read also in the meta-analysis how many of these, let's say, very basic requirements for good experimentation are not really fulfilled in many of the studies. So very often the number of animals in a given experiment is insufficient to really test the hypothesis in questions. There's inadequate controls. Very often only one sex is tested, or at least the results of only for one sex are being reported. There's unclear genetic backgrounds, or also the let's say the mutation in question has not been really clarified, and it is also known that in the lab animal strains, so the genotypes of those strains, the background strains, may themselves interfere with certain phenotypic characteristics. So this should also be recorded and also observed. And of course, there's experimental biases that are introduced by not randomizing, not blinding the experiments, and so on. The other question is about the general transferability of animal results from animal experimentation to the human situation. And here, I guess it's very clear that everybody doing animal experiments should be really clear about the opportunities, so about the applicability, but also about the constraints. And I think it's one should not exaggerate the applicability into one or the other direction. And this is shown in this slide. So this is these are two studies in the journal PNAS. And so the first group, reports that genomic responses in mouse models poorly mimic human inflammatory diseases. And then another group 2 years later redoes the study and comes to the conclusion that genomic responses in mouse models greatly mimic human inflammatory diseases. So I think that the message to take away from this is that animal models have their applicability, but one has to be, one really has to be sure about the applicability and about the constraints of the animal model used. So Björn also mentioned that study, this Friedman et al. Analysis, and I show here a slide from an update to the initial paper. And this slide is from F1000 research. So where they estimate, well, let's say that about half of the US spending in preclinical research goes into irreproducible results. And what's interesting is that they then also stratify, so which different components contribute to this irreproducibility. And this is about biological reagents and reference materials, bad or poor study design, data analysis and reporting, and laboratory protocols. And of course, if particularly as a funder, if you see such numbers and such reports and see that half of the funding that you provide is actually wasted on irreproducible results, then if you want to change this situation, you need allies in the research environment, and this is where also the research infrastructures come into play. Because the research infrastructures can, and I put this here, so this is actually taken from a brochure, a new brochure or a new information leaflet of the European Commission. So research infrastructures make science happen. And how do they do that? Because they provide easy access for scientists to state-of-the-art laboratories, equipment, biological samples, scientific data, and also the computational side of this. And of course, research infrastructures should also provide, let's say, an answer to this problem of biological reagents, reference materials, study design, data analysis, reporting, and lab protocols. And so now I would like to provide an overview how InfoFrontier is actually addressing this. So InfoFrontier is a research infrastructure. It's a consortium of, so it's a European distributed research infrastructure with 29 partners in 15 countries, and we are providing access to resources for the generation, phenotyping, archiving, and distribution of mammalian models. So rodent models, mainly mice, and also rats. One of our core objectives is to promote responsible and reproducible research. And on the left part of this slide, you see a very important aspect promoting the 3 R's: replacement, reduction, and refinement. And but in this presentation, I will concentrate a bit more on the upper right, lower right part. So minimizing duplication and particularly providing this centralized access to high quality resources. And of course, these two aspects in InfoFrontier are highly interlinked. So in terms of access to these high quality resources, so in InfoFrontier, this means model development, so the development of precision mouse and rat models, systemic phenotyping, archiving, and distribution. We provide a lot of different trainings. We provide also expertise, and then there's of course also access to the data, and all of this is centrally available through our website, is www.infrafrontier.eu. So I would like now to first concentrate a little bit on this part of the archiving and distribution, and this is done in the European Mouse Mutant Archive. So the European Mouse Mutant Archive is, or the acronym, the short version, EMMA, is the 3rd largest non-profit mouse repository worldwide. So meanwhile, we have more than 7,300 lines archived. And these lines come from individual depositors from the scientific community, but also about half of the lines come from large-scale consortia like the IMTC, the International Mouse Phenotyping Consortium, which I will also briefly talk about a little bit later. And these lines are archived in the European Mouse Mutant Archive. And since EMMA is a distributed repository, there's not one central place, but there's different places across Europe. And also the distribution of these mouse lines from EMMA is a global distribution. So our users, our customers come from all over the world. So we have about 500 distribution requests per year, and all over, we have carried out more than 6,000 shipments in total. About half of our customers are in Europe, but then also an important, so large group of, many shipments go to North America, and then also to Asia and Oceania, Australia. This shows the MStrain search. So this is how you find strains in the EMA archive, and here you can in the search window you can enter different entities if you know for instance the common strain name that you're searching for, but you can also browse by a disease category. And an example here is shown the RIP-B7, the common strain name, and you would find different information, general information about the model, information from the provider, literature references. You can also see the availability of the mouse, so either the mouse line is available to order, but there may be also other, so actually the archiving may still be under development, but then you can register interest. What I would like also to point out is this information here, the international strain designation, because we are talking about, so this webinar is about reproducibility. So the mouse nomenclature, and this is an a recent article in Lab Animal. So also having a very strict naming guidelines for these mouse lines really contributes a lot to make these research done in these mice discoverable, replicable, and this is pointed out here. And there's also tools to understand how this works, and I would like to briefly mention here this blog post by from the Czech laboratory, actually where also the, let's say, the safekeepers of this mouse nomenclature sit at the Czech laboratories in the US, and in this blog entry, and the link is here, explains how this nomenclature works. And it's needless to say that in InfoFrontier and then the European Mouse Mutant Archive, we are working very closely. So we do very strict data curation in that respect, and we're also very closely together with the Czech laboratories in this area. I would like now, from archiving and distribution, come to this aspect of systemic phenotyping, and shortly introduce the concept. So this is done in what the so-called mouse clinics. It's systemic phenotyping, this meaning that one mouse strain with a specific mutation, so one mouse cohort goes through the entire process, and then in different screening areas like neurobiology and behavior, blood chemistry, dysmorphology, energy metabolism, and so on. So it goes from the mouse line goes through all these different modules, and different organ systems and disease areas, histopathology, histology and pathology in the end, and then all the data is provided. And with this, sorry, wrong direction, what we achieve is a whole system screen of a specific phenotype, covering more than 500 parameters in different disease areas and organ systems. So you basically achieve a full characterization of a mouse model, and this provides a maximum information using a minimum amount of animals. Of course, this is really a large-scale, high-throughput operation, which has its specific logistical, experimental, and analytical challenges. And how this is addressed is, and we show this here, that the major mouse clinics in InfraFrontier, they apply quality management. And this is what I show here is the ISO certificate of the German mouse clinic, which is located in Munich. And so the German mouse clinic is certified according to ISO 9001. And well, in general, the quality management provides a structured evidence-based approach to improve quality and really to being able to constantly improve and provide the best quality to your customers. And there's different characteristics of quality management systems, which I've listed here on the left side. I don't want to now read them all, but I think the slides will be provided, or the of this webinar will be provided to all the participants. What I would also like to point out is that also in the different research infrastructures, biomedical life science research infrastructures across this, we have also developed a common view, a common framework, common principles on quality management. And these are outlined in a paper, and I will provide the link to this at the end of this presentation. So what we have shown is that InfoFrontier has a worldwide user community, and it has also worldwide collaborations. For instance, this quality management framework that I showed was developed within the context of the Corbel project. There's other projects like EASC-Live, IRDIRC, with the rare diseases community, as well as this European Joint Program on Rare Diseases. But I would like to concentrate now a little bit on the International Mouse Phenotyping Consortium, IMTC. And you see the partners here. So this is about the generation and the systemic phenotyping of mouse mutants, of knockouts for all the protein coding genes in the mammalian genome. And so the idea, and this IMPC, the International Mouse Phenotyping Consortium, is currently in full swing, is to build the first comprehensive catalog of the mammalian genome, illuminating the dark genome, because what we know is that there's very large areas in the genome where no functional annotation at all is available. So currently the IMPC has generated mutant for more than 9,000 genes in the mammalian genome, and more than 7,000 of these mouse lines have been comprehensively phenotyped. So this amounts to 84 million data points. And all this data, as well as and the mouse mutants, are made openly available. And the distribution of this mouse lines is done through repositories such as Emma, which I talked about a few minutes ago. So this is the website of the IMPC, www.mousephenotype.org, and I really encourage you to look at this website, search for genes, search for phenotypes, because by making all this data freely available, this may also help to help you in your studies, and also maybe help to avoid the duplication of certain animal experiments. So one last thing about, because we talked also about reporting, so the IMPC fully follows these ARRIVE guidelines, so the guidelines for reporting of in vivo experiments in animal research. And I also would encourage you to have a look at these guidelines. So to sum up about the reproducibility in preclinical research, I hope I could show that, convince you that all these different aspects that contribute to the irreproducibility in research are being addressed by InfoFrontier and other biomedical research infrastructures. Below here, you can see the 13 biomedical research infrastructures which work together and which are on the S3 roadmap. And I provide also the links that so this is an overview of the links that I've been showing about INFRAFRONTIER, the nomenclature, the quality management framework, IMTC, and the ARRIVE guidelines. And with that, I guess, yeah, I would like to stop here and I'll be open for questions. Thank you.
Perfect. Thank you so very much for your excellent presentation. I will now go to the questions. The first question is for Björn, is regarding the beta testing within the quality system. How long does it take to implement the quality system, and what is the first feedback from users?
Thank you for the interest. It really depends. So implementation depends on what is already present at a research institute. So it's really hard to say, because with one of the beta testers, I would say they would have within probably 1 week all items fulfilled that the system requires, and other research institutes probably need longer because they come from a lower basis. So the implementation time really depends on the research institutes and where they're coming from, and the feedback in general is positive. It's so the organizations see the advantage to have all information organized in one place, and even the universities say, okay, we need to improve the documentation, and there we have now some guidance and have some reference so that we can do that. So overall, it's positive.
That's great to hear. Thank you very much. Next question is for Michael. What are the costs and user fees involved in accessing the different services of INFRAFRONTIER?
Thanks for the question. So in general, since we are offering a broad portfolio of resources and services, so there's no one price for all. I can, for instance, point out for the European Mouse Mutant Archive. So there, the archiving itself is free of charge. However, this comes, so if you want to archive your mouse line in the European Mouse Mutant Archive, you also you consent that this mouse will, this line will be distributed as well. So we are not not only a backup solution, but archiving comes with distribution in the European Mouse Mutant Archive. If you order or request a mouse line from EMA, then it depends a little bit whether you have to order live mice or you order frozen stock. So currently the prices are at 1,600 euros for instance, for ordering frozen stock, plus shipping costs. And so this is mainly a cost recovery to fill up the repository again. And in terms of the large-scale phenotyping, so there the unit costs would be higher, and there's different access modes. For example, you can have direct collaborations with the mouse clinics, and also depending on how you want to handle the data. And so what we also did is that we provided free transnational access to access units in systemic phenotyping.
Okay, great. Thank you so very much. We have now come to the end of this webinar, and I would like to thank you all for attending today's session, with a special thanks to our lecturers for these excellent presentations. Should you still have any further questions to one of the speakers, I welcome you to submit those to us by email, and we will get back to you as soon as possible. The recording of this webinar will be made available to anyone interested on our website within 2 weeks. I would also like to point out that we will have another webinar next week, Tuesday, regarding pre-analytical validation and quality systems in biobanking. We hope to all see you again then. Thank you and have a nice day.